Thank you so much. This is the first time for me in this meeting. I'm a biologist. I'm doing basic research. And I have to say that I have never met an ME/CSF patient. So everything I know about the disease and the work that I'm doing and the will to continue working on is based completely on published studies, and that is data. So I'm not a clinician. I don't know if I can correlate some of my findings with what things that you already know. But yeah, I just wanted to acknowledge that I have never met an ME/CSF patient. The implication of viruses in ME, it's not a new concept, and we all know that many of the patients do actually mention that the trigger of the disease many times coincides with an infectious event, and the chronicity of the symptoms have been proposed to be due to the reactivation of latent viruses. And especially in the last years, this concept is gaining further and further ground. And especially we can see that from other studies, and especially from data that Professor Prusti presented just before. The effect of the viral infections or the persistent viral infections in the immune responses or in cellular physiology have been described before, and I will not go into the details of that as they have been described in much more detail from an expert in the field. We are interested in the herpes viruses, and the herpes viruses are infections that are found in the majority of the population, where they persist lifelong existence in the host. However, those viruses, they do reactivate from time to time, and the purpose for the reactivation from the virus perspective is to ensure its persistence, to show that it can infect uninfected individuals and it can continue to live on. So this reactivation, though it may occur spontaneously, in most cases it is triggered by a specific stressful event, and that event can be an infection, a septic shock, or trauma. Now, all this is happening both in healthy individuals and in patients with ME/CSF. But given that there is a very specific background of immune suppression in ME/CSF, of immune incompetence, that may be the reason that that may explain the chronicity of the symptoms in the patients. Another form of latent viruses are the human endogenous retroviruses. And these are retroelements that we have acquired during evolution. They actually constitute a significant amount of our genome that goes up to 8%. And though these are replication deficient viruses, the transcriptional activity of those elements has been evident. And they have been proposed to be induced either by infections or inflammation, again by another stressful event. The potential functions of those viruses, they have been proposed to regulate gene expression. They have been involved in antiviral responses and placental formation, but the deregulated expression of those elements have been associated with diseases, and specifically with cancer and neurodegenerative diseases. Following the COVID pandemic, now we know that SARS-CoV-2 infection can also induce the transcriptional activation of those elements. And there are evidence from analysis of bowel fluid and bowel fluid cell samples. We now know there are multiple studies that confirm that SARS-CoV-2 infection can indeed reactivate latent viruses. Most of the studies came initially from patients that had the severe form of the disease and that they were either hospitalized or they were treated in the intensive care unit. So all herpes viruses, including EBV, HSV1, CMV, varicella, HHV6, and the latent endogenous retroviruses have all been found to be reactivated, or for the herpes transcriptionally activated. And this reactivation, in some cases, it has been proposed to account for the prolongation of the patients in the clinics and the difficulty to recover. Now, the severe COVID fortunately affects not that big proportion of the population. So what most of us have had, it was like a milder form of the infection or even an asymptomatic form of the SARS-CoV-2 infection. So one of our first objectives was to check whether a mild form could actually reactivate the latent viruses. And if so, what it is, does it differ? Is it the same in the case of the ME/CSF patients? And nowadays we know for long COVID. When we first started, when we were preparing our study, there were very little evidence about the reactivation of the herpes viruses in long COVID. But since then, a number of studies have been generated that they do show that there are indications for the reactivation of the herpes viruses in those patients as well. And primarily it's EBV, but also it's HSV6 and HSV1. In some cases, in one study has shown that the EBV, the antibody responses against EBV, they correlated with symptoms like fatigue and neurocognitive dysfunction. Whereas in another one, the responses against EBV and varicella, they correlated with T-cell effector memory functions. But I guess we will hear more detailed talk about this in a while. So the objective, the initial objective actually of our study was to check whether the reactivation of the herpes viruses differed between healthy donors and ME/CSF patients. However, since the time that we performed the sampling process was in spring of 2020, inevitably we had both patients and healthy donors that have been infected with SARS-CoV-2. So that actually gave us a unique opportunity to have now our own trigger for the reactivation of those viruses and see if the responses differ. It happens many times that we don't see things at baseline or at rest. This is something that we also do in the lab. We need to trigger, we need to give a stimulus in order to check whether the downstream processes differ or not between the patients and the healthy donors. So we have analyzed for a panel of antibody responses for a panel of herpes viruses, specifically EBV, HSV6, HSV1 and 2, cytomegalovirus, varicella, and the herpes. And in the second study, we have also analyzed antibody responses against the human adenoviruses. So we would like to see whether are there any difference between the healthy donors and the ME/CSF patients. The very first thing we needed to do at the time was to confirm which participants have actually had the infection or not. But again, as I mentioned earlier, since we did the sampling in June 2020, the PCR tests were not available at the moment, so we needed to find a way to somehow define which of the participants have had the infection and which not. So we have initially analyzed the IgG responses against the receptor binding domain for SARS-CoV-2 in the blood, and we have used a cohort of blood donors from 2015 as controls to define which one of those were positive and negative. Next, we did analyze the saliva samples from the same patients, and the interesting observation that we made was that many more patients had high levels of antibody responses in the saliva that were not present in the blood. Now, in order to define, again, which ones were positive and not, this study is actually a longitudinal study. So we had multiple samples from all these individuals every 3 months. So we used samples from a specific subgroup of 19 patients that were negative when they were initially recruited in the study, but following after 3 or 6 months, they came with a confirmed PCR test or specific symptoms for SARS-CoV-2. So overall, this was a bit of a surprise of why there are some people that have so strong responses in the saliva, they do not have it in the blood. And we have attributed this to be most probably in the people that have the antibodies in the saliva, most probably were asymptomatic and they didn't know they have encountered the virus. So, but since we wanted to deal with this in the most proper way, we have segregated our cohort into systemic and local responders in order to check whether there are any differences there. And as you can see here, oops, we can actually see that the antibody responses against COVID, they were actually higher in the ME patients compared to the healthy donors, both when we compared the local responders and the systemic responders. And similar finding we found for the IgM subclass, which the IgM antibodies give, let's say, an indication of a more recent infection. No differences were noted for the nucleoprotein, though. So since we have done this segregation between systemic and local responders, then we evaluated antibody responses against this panel of herpes viruses. And for EBV, we have specifically analyzed VCA and EBNA1. And what we have found was that specifically for EBNA1 is that following COVID infection, either in the local or the systemic responders, there was a significant upregulation of the antibodies there. And that was something that we could not see in the healthy donors. So EBNA1 immune responses were only found in the ME patients. When we checked for the viral capsid antigen, though this was not something unique for the ME patients, still we can see that when we compared the negative versus the either systemic or local responses, the significance of the upregulation was much higher compared to the healthy donors. Now for HHV6, excuse me, we had similar findings with EBV. So again, the antibody responses in the ME cohort, they seem to be more strong compared to that found in the healthy donors. And this was actually true both for the IgG and the IgM. Similar finding was found for the endogenous retroviruses, and even though, again, this was not something unique for the ME, however, we can see that the differences in the antibody responses were stronger in the ME patients. Now, in order to confirm that what we're doing is actually valid, we have also investigated the responses for the latent viruses in this cohort of 19 patients, 19 participants actually, that I mentioned before that we used for the evaluation of the positivity of the SARS-CoV-2 antibodies in the saliva. So we did the pairwise analysis of the same individuals before and after the COVID infection, and we can see that indeed the antibody responses go higher within the same individual. Now, The interesting thing is that we have done all this in the saliva, but we have done it also in the blood samples. And when these are just some representative graphs from the analysis that we have done regarding EBV or HSV1, and what you can see specifically for VCA is that when you analyze the blood, you get nothing, like no differences, no responses. Similarly, for the antibody responses for COVID, even though we do see, yes, in the positive groups, there is an upregulation. However, the differences that we saw between the ME patients and the healthy donors is again lost. So any differences that we see in the saliva, we cannot actually detect in the blood. And this is just a summary of what I've been describing so far. So we can see that following SARS-CoV-2 infection, the antibody responses against latent viruses, as we can see, is much more pronounced in the ME. And specifically for EBV, EBNA1, which is unique in the ME, VCA, but also HHV6 and HERV-K. So now I would like to go to the second study that was recently published, and that involves the antibody responses against the human adenoviruses. A collaborator from our lab was very interested in investigating those immune responses in ME patients. And the human adenoviruses are, again, they are infections, they're typically mild infections that they affect the respiratory, gastrointestinal tract, or the conjunctiva in the eye. The adenoviruses are double-stranded DNA viruses, non-enveloped, and there are more than 110 serotypes known, which are classified into 7 different species. The tropism of those viruses differs depending on the serotype. And again, in individuals that are immunocompromised, they're again at higher risk of a persistent infection that may occur depending on the tropism, either in the tonsils, the adenoids, the gastrointestinal tract, or other sites. And what we saw was that, again, similarly to the other herpes viruses, for the adenoviruses as well, that the responses were much stronger in the patient group compared to the control. And actually, we could not find any reactivation in the healthy donors. So this was something, again, unique in the group of the ME patients. And when we analyzed also the blood plasma, again, regardless of this, we could find absolutely no differences when checking the blood. Yes, so just again to confirm, in the same batch of, in the same subgroup of the participants, that the antibody responses are actually due to the COVID infection. So just to summarize, we have seen that the mild or an asymptomatic SARS-CoV-2 infection can elicit antibody responses against herpes viruses, and more specifically EBV, HHV6A, and HERV-K. And that the responses against EBNA1 and the adenoviruses are unique in the patients. HHV6A and HERV-K, though not exclusively in ME patients, the antibody responses were much stronger in the patients. So we took this great advantage of having a SARS-CoV-2 infection as like an experiment of nature to check how an external trigger, how an external stimulus does affect, or how the patients respond, and if they respond differently compared to the healthy donors. And the second, let's say, sort of the second observation that leads us to the same conclusion is that this antibody signature that we see in the saliva is absent systemically, and this would come as no surprise, I would say, and this complements, I think, what also Professor Prusti was saying before. So the tropism of these viruses in specific tissues implies that maybe we should be using samples that are more relevant or more local to the site of the reactivation of those viruses if we would like to see differences. So I would like to thank you all. All these experiments were done with samples from the Brageck Clinic and from patients in the region of Stockholm, our funders, and thank you for your attention. Thank you so much.
Thank you, Dr. Apostolou. And again, actually, I think we have time for a few questions. Yes, please.
Ni kan prova att använda mikrofonen vid er bänk, se om den funkar.
Perfect.
Tryck på mic-knappen.
So I have a question regarding actually the healthy donors. As I have quite a few patients who suffers from post-COVID and CFS after vaccination, I wondered, did you take that into account, looking at the healthy donors from that perspective?
The samples that we have used in these studies were like from almost 3 years ago. There was no vaccine available at the moment, so none of them is vaccinated. And it was also too soon to have healthy donors that could potentially, after the infection, would develop long COVID symptoms. So, no.
Okay, so they were sort of vaccine naive. No, I just find that interesting because I have quite a few patients in that group, and it would be interesting to see whether or not it's the same kind of development of disease behind it.
Yes, absolutely.
Good, thank you. More questions to Dr. Apostolou? No questions from in here. Again, I'm not sure if we can get any questions from the online viewers. No? Not right now. It's been a long time since I worked with human herpesviruses. But what about cross-reactivity? When you see these antibody responses to several different herpesviruses. These antigens, they are very unique to each and every one of them, I guess.
Yes. And I mean, we have done also HSV1, HSV2, cytomegalovirus, but we didn't find any differences there. So these EBV, HHV6 were the two viruses that they stand out in this analysis.
Very interesting. Thank you. Okay. So thank you, Dr. Apostolou.